So the Doheny Beach Molecular Source Identification Study is part of a larger study that we're conducting for the state called the Source Identification Pilot Protocol Project. And this study is designed to develop protocols that can be used throughout the state for microbial source identification. In this case, Doheny has long been one of the most contaminated sites in California. It's been the site of a major epidemiological study, and was found that people swimming there still became ill even when no bacteria was present. So the thrust of this study is to actually develop methods that can be used throughout the state to identify sources of bacteria at beaches. We're mainly looking for the sources of bacteria at this particular beach. And that each beach is different. This beach, primarily the two main sources that we can see just observing are humans and seagulls. And so those are the most obvious things to look for. But at other beaches, there may be agriculture in the area, there may be inputs from livestock farming, there may be inputs from agricultural fields. And when the creek is open at this beach, then one of the major sources is the storm drains. But during the dry season, the creek isn't flowing, and so the major sources are either seagulls or humans as far as we know. And so that's why we're testing the sewer system, is to kind of eliminate that it's actually infrastructure that's causing the problem. So there are two different sampling periods with this study. The first is weekly. So once a week we've been collecting samples up in the storm drains and along the beach. And then the second is this 30-hour study. And so you see we've set up a motor home so that people can catch a little bit of sleep, but mostly we're collecting samples for 30 hours around the clock. So we've set up labs actually in the parking lot on the beach, and we're actually processing the samples on site. At this study we have three different types of sampling going on. The first thing we're doing is looking at groundwater. We've introduced dyes to all the toilets surrounding the beach, and so we're looking to see if the dye is coming out through the groundwater. If that is the case, then what we will see is we'll be able to measure that dye in the ocean water and the groundwater. The second thing we're doing is actually looking for bacteria. We're sampling both on the beach and up in the watershed, and so we're testing the storm drains both with culture methods where we measure live bacteria, and we're also filtering and freezing bacteria on filters for later analysis.
So during the 30-hour study, we performed water quality analysis on all of the sample sites by using two of these more traditional culture methods. The first culture method we did is called MEI, and we filter a known volume of water through a filter and put that filter onto an MEI plate that has a growth media. MEI targets enterococci bacteria, and we put that plate into an incubator for 24 hours. 24 hours later, we count how many colonies formed on that filter, and with that information, we're able to calculate what the concentration of bacteria was at that site at that sample time. So the second culture method we employed was called co-alert, and that targets E. coli. And for that, we take a known volume of sample water and pour it into a vial that has a liquid growth media in it. So you mix up that and pour it into something called a quantatray and seal that. You put it into the incubator, and 18 hours later you count basically the amount of change in color and the amount of fluorescence under UV light. And with those two, and with that information, you're able to determine what the concentration of E. coli was at that site at that time. In addition to those two more traditional culture methods we employed, we also filtered larger volumes of water through filters, rolled up those filters and put them into a microtube and put them in liquid nitrogen to flash freeze them. So once they were frozen, we're able to archive them, and at a later date we're going to be conducting multiple qPCR assays on them and basically using different molecular techniques and molecular methods to determine what the source of this bacteria is, because we've already used traditional methods to show that it's there.
When we get the results of another study we're conducting, which is an evaluation of source identification methods, then we'll use the best methods from that study to analyze these filters. And those are sources such as humans, seagulls, livestock, and other waterfowl. Once the results of the analyses are in, then those will be used to help managers determine the best way to remediate those sources from the beach. The regulators look at levels of bacteria at the beach, and regardless of the source, if these particular groups of bacteria exceed thresholds, then the managers, be that the city or the county where the creek flows, are asked to do remediation to stop bacteria from the storm drain or the creek from entering the beach water. And the reason that we're concerned is that the worst case scenario is that you have sewage flowing to the beach. And so these bacteria that we measure are always in sewage, but they're sometimes there when there isn't sewage too. So that's the main reason that we're doing these source identification studies, because it could be we have bacteria growing in the environment that aren't associated with pathogens. And it's a much different scenario than when you have sewage, where you have human sewage and you always have human pathogens. And in that case, you can almost guarantee that people will be getting ill. The goal of the source identification pilot project is to develop a manual that can be used by municipalities and others that are charged with cleaning up their watersheds to do a better job of developing studies that will lead to a successful implementation and remediation of their contamination problems. There's really there's no set protocol for doing these kinds of studies, and so we're doing studies throughout the state at multiple different kinds of watersheds in order to develop a manual that will give people guidance when they need to do these kinds of studies in their watershed.